So why will a SARS-CoV-2 vaccine take a long time to be created, and how would it actually help? Well, in order to answer that question, it's important to understand how viruses and vaccines work within the human immune system. Viruses are little more than genetic material covered in a protein coat. Viruses require a host cell in order to replicate. The SARS-CoV-2 virus is a coronavirus characterized by its spherical shape and glycoprotein spikes. Its genetic material consists of a single strand, RNA, in a helix shape. The virus's spike glycoproteins are recognized by receptors on the surface of the host cell. Once the spike protein is attached to the receptor, the virus is pulled into the cell through a process called receptor-mediated endocytosis. The virus then releases its RNA genome inside the cell. The RNA is replicated and read by the host cell, tricking the cell into constructing viral components. New viruses are assembled from these components and are released from the cell via exocytosis. Vaccines are substances that stimulate a response from the human immune system in order to prepare it for possible infections. This involves introducing an infected pathogen, or a fragment of the pathogen, to the immune system. The pathogen is recognized by the immune system as a foreign invader. White blood cells, known as macrophages, destroy the invader. The macrophages keep a piece of the destroyed microbe called an antigen and pass it on to memory B cells. The memory B cell remembers the antigen so that it can recognize the invader if it ever returns. When the memory B cells encounter the exact same antigen again, they produce large amounts of proteins known as antibodies. The antibodies attach to the antigens they recognize and call over macrophages to attack the intruder. This is your basic adaptive immune response. Basically, a COVID-19 vaccine just needs to present part of the SARS-CoV-2 virus to the body, and the immune system will do the rest. Sounds simple, right? So why does it take a long time to actually make a vaccine? Well, vaccine development is composed of several steps. First, researchers determine how the vaccine should trigger an immune system response. Then, they start testing their hypothesis in a lab to make sure that it works. This usually entails years of research, experimentation, and peer review. At the moment, there are many different vaccines for the COVID-19 disease under investigation, each using a different viral protein to stimulate the immune response. Some labs are using RNA in their vaccines to have the body itself construct the spike glycoproteins found on the surface of the virus. The next step is human testing. These trials are conducted in phases. Researchers start with phase 0 testing. The number of participants is limited, usually consisting of around 10 subjects. The vaccine is administered to the participants in small doses. The purpose of this phase is to determine if the drug is safe enough to continue with testing. In phase 1, the vaccine is given to a group of less than 50 people. These people are then monitored for the expected immune response and for any possible side effects. Phase 2 involves giving the vaccine to several hundred participants. The scientists continue to monitor for safety and determine the proper dosage size. Phase 3 is conducted with thousands of participants. The goal of this phase is to gather statistical data on the effects of the vaccine on a large population. After all the testing, the new product must be reviewed by the FDA and the CDC for licensing and safety regulations. Phase 4 is conducted after the vaccine is approved. The new vaccine is monitored to confirm long-term effectiveness and determine any side effects. Normally, this process can take years to complete. However, because of the similarities between the SARS-CoV-2 virus and the 2002 SARS-CoV-1 virus, vaccine development is actually progressing at a relatively swift pace, with many proposed vaccines currently in phase 1 testing. This process may seem very slow compared to the rapid spread of the current viral pandemic, but testing has a purpose. Without proper testing and review, a distributed vaccine can have unintended consequences. For example, an RSV vaccine was given to toddlers and infants in 1967. They later experienced an enhanced form of the disease when they contracted the actual virus. This is known as antibody-dependent enhancement, where improper attachment of antibodies to the virus actually makes it more difficult to remove. And because of this, 2 infants actually passed as a result. A more recent example of this phenomenon is detailed in a 2004 report on SARS-CoV-1 vaccine testing on ferrets. The report explains that upon exposure to the SARS virus post-vaccination, the ferrets developed severe liver inflammation. Testing and observation are essential parts of vaccine development. It may take some time, and the pandemic will likely slow down before vaccines are ready for mass production. It is also important to understand that vaccines are a preventative measure, not a cure. Our foremost priority should be not to get infected in the first place. Remember to stay safe and be patient, and make sure to wash your hands with soap and water for at least 20 seconds every day. Thank you.